In the quiet dark of a biology lab, a tiny flash of light can signal a breakthrough. This spark is not from a microscope or a computer screen, but from a molecular beacon known as Luciferase, derived from organisms such as 𝗘𝗫𝗣𝗟𝗘𝗏, the common firefly. Luciferase enzymes catalyze a bioluminescent reaction that has become one of the most sensitive and versatile tools in molecular and cellular biology. The science behind the spark. Luciferase is an oxidative enzyme that catalyzes the conversion of a substrate, commonly known as luciferin, in the presence of ATP, oxygen and magnesium ions into oxyluciferin, emitting visible light in the process. The amount of light produced is proportional to the amount of enzyme present, making it an ideal reporter system. There are several luciferase systems, including firefly luciferase, 𝗘𝗫𝗣𝗟𝗏𝗏, which emits yellow-green light around 560 nanometers. Renilla luciferase, Renilla reniformis, which emits blue light around 480 nanometers, and NanoLuc luciferase, 𝗕𝗢𝗪𝗟𝗘𝗏 gracilis, a small engineered enzyme with bright and stable luminescence. Each has unique properties that make them suitable for different biological applications. Applications of the luciferase assay. One of the most common applications of luciferase is as a reporter gene. By linking the luciferase gene to a promoter of interest, researchers can quantify the promoter's activity by measuring the emitted light. This is particularly valuable in transcriptional activity assays, enhancer, promoter mapping, and signal transduction pathway analysis. For example, in cancer research, luciferase reporters are used to study how transcription factors like NFB or P53 regulate gene expression in response to stress or drug treatment, drug screening, and high-throughput screening. Luciferase assays are favored in HTS due to their high sensitivity, low background, and rapid readout. They are used to screen for inhibitors of viral replication, modulators of transcription factors, and enzyme inhibitors. In antiviral drug development, a luciferase gene can be engineered into a viral genome to monitor viral replication dynamics in real time in response to compounds, protein-protein interaction studies. Bioluminescence resonance energy transfer, BRET and split luciferase complementation assays enable the study of real-time protein-protein interactions in living cells. This is particularly useful in G-protein-coupled receptor research, where receptor ligand and receptor effector interactions can be visualized and quantified. Cell viability and cytotoxicity. Luciferase-based assays, such as cell titer glow, use ATP as a proxy for metabolic activity. Since ATP levels decrease in dying or stressed cells, luminescence serves as an accurate measure of cell viability. This application is widely used in toxicology and oncology to evaluate the effects of experimental drugs on cell survival. In vivo imaging. Luciferase is used in bioluminescence imaging, bleed to monitor gene expression, tumor growth, metastasis, and microbial infections in live animals. The emitted light can be captured using sensitive CCD cameras. Luciferase-tagged cancer cells implanted in mice allow longitudinal tracking of tumor progression without euthanizing the animals. Advantages and limitations. Luciferase assays offer several advantages. They are extremely sensitive down to femtomole levels, non-radioactive, safer than traditional radioactive methods, and well-suited for high-throughput and real-time analysis. However, there are some limitations. The signal can decay over time depending on the luciferase type. Substrate delivery and timing must be optimized, and cell lysates or test compounds may interfere with the assay. Conclusion: lighting the way forward. Luciferase assays have revolutionized molecular biology by turning invisible biological processes into visible light. Their simplicity, sensitivity, and versatility have made them indispensable in research ranging from basic gene regulation to cutting-edge therapeutic development. As synthetic biology, gene editing, and in vivo imaging continue to evolve, luciferase will remain at the forefront, illuminating the inner workings of life, one photon at a time.